from last 40 years, when I was research scholar, like, I think, yeah, 84, 85 onwards, when they conceived the idea of Human Genome Project, 1985, by Robert Sinsemeyer of Institute of California, Santa Barbara, and he made a like-minded fellows. Then he put forth that can you sequence the complete genome and address the problems of genetic diseases, of which runs about 4 to 6% in the humanity. From then, that time, as a research scholar, I started reading that whatever is available to me in the library, and updated I used to make it. So that made me my first talk to get an idea about what is being done in the field of genomics in last 40 years also. This is the outline of my talk. First, genes and genome, I introduced. Some of the things are already known, but I wanted to put it in a perspective manner here. And after the Human Genome Project, what has happened for the genome and in the post-genomic era, then we should also aware how genomes are evolved in metazoans or the eukaryotic system from prokaryotes, and some scientific breakthroughs. I hope many of you know, but still I thought I could make them also. Then I'll conclude that where we stand in the genomics. Can you read this? Generally, I use this first slide many a time. Maybe last time also, I don't know, I put it here also. So, without this slide, I cannot go further. What is the answer? Genetics, genes, or genome. So, I will not argue with that if somebody says that genetics or gene all are synonym or they're close, they're together. So, we are here, or we look like ourselves because our genes and the genome is responsible. At that time, when that interact with their cellular environment, that's also more important. So whenever I go to talk here, all these aspects, 50% is the genetics, 50% in the environment. Environment also responsible. I'm not telling you everything is by genetics. Cellular environment as well as our physical environment also responsible to make ourselves as we are today. I hope you know what are genes. They are the basic unit of everything, and they determine the nature and function of the cell, and the unit of genetic informations. They control the phenotypes and they synthesize the polypeptides, and the genes encode protein, which is, as you know, required for growth and maintenance and differentiation and development. And Johnson, 1909, coined the term gene, and the other contemporary scientists were rediscovered the Mendelian principles, and William Bateson took a lead and to translate from the German language of Mendel work into English work and publish the principle of genetics, the book. That time, the Johnson coined the term the gene. That was very appropriate at that time also. I hope you can see this image. Can you make some of this characteristic feature? Or someone you can touch your earlobes. It is attached or free. We can have both here. So even the dimples, some of the persons are having dimple or cliff chin. All these are nothing but the traits, characteristic features we do have. They are controlled by genes. The traits, whatever you have, some phenotype just I recorded here, and many of them are like the last one, allergy is a multifactorial. Many things are involved there, but some of them are curly hair. All these are controlled by fairly a single gene there, and most of them are dominant characteristic features there. So all these traits are controlled by genes, And look at the last images here. We are made up of from our grandparents, maternal, paternal, grandparent genes. They also contributed. Our parents' genes, father and mother, and also we share our genes from brothers and sisters. And fourthly, we have our unique characteristic features of the genes combination, because every egg and sperm produced from our parents are unique recombinant products. So they have undergone fusion, the zygote formation, fertilization, then they have undergone that many recombinations also, and that is making us unique. So we share with our ancestor, but also we do have our own unique kind of the characteristic features. So what is the genome? Total DNA of an organism, including the coding and uncoding sequences. Majority of the organisms have the DNA as a genetic material, and some viruses having RNA as a genetic material. We do have many kinds of genomes. Generally, you can classify them. Prokaryotic genomes, where bacteria, archaebacteria put together. Eukaryotic genome, including plants and animals. When you say eukaryotic genome, all the eukaryotes. And nuclear genome, only the genome present in the nucleus, and mitochondrial genomes, mitochondria present in plants and animals, and the DNA present in the mitochondria, and they are called mitochondrial genomes. Mitochondria are present in many copies, in particular mitochondria itself. Not like one single copy like in bacteria, like in a single cell organism. Chloroplast exclusively present in plants. What is genomics? Simple study of genomes. How? Using set of techniques, analytical methods, and asking n number of scientific questions. And this makes the complete study of the genome. The genomics is the study of the genetic makeup of an organism. It employs the recombinant technology, DNA sequencing methods, bioinformatics to sequence, assemble, analyze the structure and function of the genome. All this put together, the field we call as a genomics. And also we have structural genomics, functional genomics, comparative genomics, and so on. So not many other classifications can be done, but to understand the genome and study of the genome. Why should we study genome? Don't tell me that we are not studying, sir, because your syllabus is not there. In your syllabus is not there, so you are not studying. So why do you ask the question? Why you had to study genome in this era?
Targeted medicine.
Yes.
Correction.
You are right. But now, why? Only our genome is made up of 4 nucleotides. It be bacteria, or animal, or human, or plants, all are made up of 4 nucleotides. And these 4 nucleotides in all permutation, combination make different kinds of protein, different kinds of organism, and different transcript they can also make. They have a hidden code determined which protein should be made when in particular tissue or the cells. And the language is common for all the organisms. Any change in the genome leads to abnormality broadly, or disease in plants, or even the organism may not be able to survive. The lethality can be there, or some abnormality will be there. Any change in the genome. I hope all of you are aware of sickle cell anemia.
Yes.
How does it cause? Because of change of one nucleotide in beta chain, and that leads to one amino acid change. Generally, our blood cell is oval shaped, become single chain. Just one nucleotide chain, one amino acid sequence. It becomes a drastic change. If both of the genes in diploid organism, the individuals are homozygous, there are a lot of severity, they may not survive also depending on this, the homozygosity, this single nucleotide and single amino acids. Okay, that's why the abnormality extend to mild, or even it will not going to cause any kind of the defect, and to the severity and other extreme also. The whole humanity is about 6% of the genetic diseases, and means out of 100 individuals, 6 individuals are suffering from genetic diseases, maybe in the beginning of the birth, or late age, or young age, or late age, this disease are there. And especially even the Indian genome scientists put forth that not only the Karnataka and Andhra having consanguineous marriages are more of the inbreeding, even the whole Indian genome itself inbred genome. So looking at the features for several generations, because highly committed to their family, committed to their community, and that's there. So more of Indian genome, individuals are inbred individuals. So if they inbred, the more of the defective genes, if they are in homozygous conditions, if they are recessive, they appear. If they are dominant in heterozygous condition, they appear. So many diseases, with the advent of technology, we are recording now. People might say that earlier there was no, they have not identified this kind of diseases. Now they identify because the technology is available. So diagnostic tools are available to find out the genetic defects. To address the 6% of the abnormality in human population, as I already told you, like-minded people 1985 conceived the idea, why can't you do some complete genome sequencing, identify the culprit gene, and they can think about that, thinking that their things are very simple, every characteristic features are caused by a single gene, and that was their hypothesis, and we can pull out the gene, or we can modify the gene, or we can make a treatment for the gene, so that way the humanity will become better, human welfare will be better. That was the idea in 1985, and they started with a lot of opposition from the other group. I am not going to go into the more history about that. Then finally they conceived in 1988, and they made James Watson as the first director of the Human Genome Project in 1990. They started, and they planned for 15 years for 5-year 3 phases, and the Human Genome Project. Because of the advent of technology, the whole project has completed, started in 1990, completed 2003, 13 years. What are the objectives? In the beginning, the objectives were only 3: completely sequence, and they understand the physical phenotypes, the associated phenotypes, and the associated genetic map and physical map, and completely sequence. Later, it has also been refined that identify all the genes, determine the sequence, store the informations in the databases, and also parallelly the databases also improved, and the technology improved, and develop the tools for analyze the data, and also the last one is very, very important. If the people identify, say that this family, this group, this community has the disease, and or this individual has the disease, and they will not get their bread and butter, they will not get job, and so address those ethical, legal, and social issues also. So they made a legislation for this. So they started, and 2001, Bill Clinton was American president. He brought together the public funded and private funded, where Craig Venter started the Celera Genomics of the private company. He was in the NIH in 1998. He came out of that and he started telling that the whole world that I am going to sequence the whole human genome in 3 years with his own technology. he did it, and he brought public funded and private funded together, and they published the working of the human genome sequences. Public funded sponsored one in 15 February 2001 in Nature, and one day later for the private funded, the Silarajoni, the complete data, working of data in Science Journal. And April 14th of 2003, they announced that we have completed the human genome sequence, 99% of the human genome sequence. To commemorate that, for 50 years of DNA double helix structure discovery. So they made the date that after 50 years of discovery of double helix structure, we now sequence the human genome. And the kind of work what they did was very systematic manner, isolate individual chromosome from bulk gel electrophoresis. Individual chromosomes are purified and cut into different fragments by restriction enzyme. The rare cutter restriction enzyme, each fragments are cloned into small vectors. Later they identified the artificial chromosome, they cloned into artificial chromosome, larger segments, and systematically each segments have been cloned, annotated, and combined that also for each chromosome and for all the 23 types of the chromosome. It took about 13 years also. That's a systematic way. So, whereas the Craig Venter did a different manner called as shotgun method. He fragmented the DNA, taken each fragment and cloned into vector, and he has a very powerful computer to identify overlapping sequences. Then he made in 3 years what these people have did in 13 years, 18 countries, more than 30,000 laboratories are involved in this. So finally, the complete genetic component, you can see here that they identified 1.5% is the coding for the gene, and about 26% is the introns. I hope you know what is introns and exons. And about 20% is the line sequences. Line is nothing but long interspersed nucleotide elements. They are the jumping genes. Line, the human has a L1 jumping genes in human. The live one we have, and that's about 6.5 kb sequences of the jumping genes, and about 13% of the signed small, about 300 nucleotide repeated sequences are there, and with the left and right arm are homologous or they complementary to each other. Like that, they also have many repeated DNA sequences, transposons and simple sequences. They are also identified in the human genome. So the coding sequence is only 1.5 to 2%. With this information, they say that with other sequences, 99.6% of the DNA between us or among us is identical. How much? 99.6%. In some literature, some article they may say 99.4, 99.5 or 6. So take it broadly about 99.6%. So how much is different? Here 0.4% different between any two of us here. Not only here, globally. So which amounts to 4 million nucleotides were different between us. Let us keep that in mind. So only differences of 4 million bases. I'll come to that later also. So they have the Human Genome Project mapped the genes and the total DNA present in every chromosome. The first chromosome in the cytologically is the longest chromosome, is having about 279 MB sequences, about nearly 3000 genes. That way they, based on the size of the chromosome, they cytogenesis are arranged about first chromosome to the 22nd chromosome and X and Y. So if you look at after human genome sequencing, 8th chromosome and the 8th chromosome and the 9th chromosome here. 8th chromosome is it's bigger than the 9th chromosome, but however genes are more than that. Look at the 11th here, the chromosome 11. naturally, cytologically smaller than the 8th or 9th or 10th, but the number of genes is almost double, indicating that the size of the genome has nothing to do with the number of genes. Okay? Similarly, they also found that the 17th and 19th chromosome and 18th chromosome. 18 and 19, if you compare, 18th chromosome has 86 MB, but only 700 and odd G, but the 19th chromosome have over 14,400 and odd genes there. And I hope you know what is the role of the 21st chromosome. Any idea you have? What is known for the 21st chromosome? Trisomy. The Down syndrome is a 3 copies. It has about 300 genes there. 45 MB and 300 genes. And now the scientists have latest article in the Cell that has come 2 months, 8 months ago. They critically identified the gene which the gene is responsible to make the heart diseases in the Down syndrome. They identified gene in mouse model, creating a transgenic mouse model, and they pinpointed 1 or 2 genes are responsible to make the defective heart in the Down syndrome babies. Recently, I made a review on that and I made a prospective article on that, which is going to publish very soon. That means not the whole chromosome is a critical region. They said 8 genes, they said in the beginning, now 1 or 2 genes are sufficient to cause the heart defect. X chromosome, where the female has 2 chromosomes, there are 1184 genes. Y chromosomes, a tiny chromosome, is responsible to make the maleness, having 231 genes. To compile the whole human genome data, what is the smallest gene? What is the size of the smallest gene? I thought my students are very sharp. I put there in the my data there, but some of you might have seen that, right? The smallest gene is 22 nucleotide. That is microRNA, which is very familiar now. We got it. The longest gene, I hope you are also aware of it, is present on X chromosome, called dystrophin gene, is about half the genome. 1 gene of the human gene is equivalent to half the genome of bacteria. What is the bacteria? The Escherichia coli sequence is about 4 MB. Here 2400 KB or 2.4 MB is the 1 gene sequence, is the biggest gene, longest gene known so far. Dystrophin is called muscular dystrophy. And there are many specific to human. There are human specific antibodies, T-cell receptors, antigen for major histocompatibility gene, and cell signaling for cytokines and blood clotting and apoptosis factors are very specific to human. They have been identified. So, while the people are compiling their human genome data and they are announcing that, the scientists, contemporary scientists, they ask questions. The Human Genome Project is not going to resolve any issues of it because it's a cocktail of 30 individuals and that they have not been revealed and is not responsible or not representing any kind of ethics group. It's new this. The people have committed it and they started the mini genome project later. They asked different kind of questions. So now we should go for like this. So there are many genome projects they launched, and I just quote some of them are also. First the International Heart Project they launched, then the Individual Genome Project, 1000 Genome Project, ENCODE Project, Synthetic Genome Project, Personal Genomic Project, One Lakh Genome Project, Genome Editing, Gut Microbiome and Pangenomics. And finally, very clever Indians started Genome India Project. When the Human Genome Project started in 1990, we are not there in the scene. So our economy, our conditions were very, very bad, but still our scientists were looking for that. And now, after the human genome sequencing announced that we are not interested in structural genome, we are interested in functional genomics. So many groups in then we are started working in functional genomics out there from the structural genomics, and lately I'll go to review some of them also. India also started from a rare diseases genome project where CCM is majorly involved. So after the human genome project, the individual genome project, the James Watson's DNA itself have been sequenced for $2 million the expenditure from this two company, this 454 life science company, and they gave 2 DVDs to Watson that this is your DNA sequences also. Watson said that, yes, I'm happy that I'll take it, but don't reveal some of the things of what apolipoprotein gene, they not to be revealed. When he said that, everybody said that, Watson said that, not to tell the apolipoprotein gene, what is that, let's go. So, everybody knows that he has a problem with the Alzheimer's disease, and he told the scientist, everybody quoted, and everybody known about that. Many times he said that, don't tell to anybody, you tell that. So everybody said to the other person, transmit in the same manner. So it did happen. So 3 billion base pair, they took 13 years for spending $4 billion, but 6 billion base pairs of Watson DNA they spent for 2 years and $2 million. Craig Winter, he had his own cellular genomics, very crazy scientist, very upcoming, very dynamic scientist. He sequenced his own genome, published in September 2007, and his genome size is only 2.8 billion base pairs. Whereas the average genome is 3.2 base pair, and his genome is so more of deletion he could see in his own genome, and also found many variations which was not found in the human genome project. And he identified the risk genes there, Alzheimer's, heart disease, and muscular degeneration, and he started from 2007 onwards starting for cholesterol thinning. It was there in his own article also. He mentioned that also. So because otherwise I will have a arteriosclerosis, the blockage of the heart arteries will be there. So he started taking that also. They said that. So and he after sequencing that, see, I have sequenced my own genome. I know the defective genes, and also I am taking the medicine. So this is the model. So then we can go for the genomic medicine for the individuals. That was one of the standard. So I would have got this slide also that before those the individual, but after completion of human genome project, even before completing, even after completion of the working group of human genome project, scientists have started asking that it's not sufficient, we are not getting anything there. Then they started international HAPMA project, about 270 individuals of 5 ethics groups of different nations, the China, Beijing, UK, and there's Wickham Shuttle, UK, and NIH group in Canada, and Japan group, using this two companies, 454 and Applied Biosystem of Illumina. This company joined together, wanted to identify any culprit variations in the genes in these 270 individuals, and also the population they selected are the trios of parents and the one individual here, trios of this, what is the Central Europe, Japanese here, and even African 30 trios. They made total 270. Later they added other individuals from different ethnic group, and finally the finding is that they identified about 170 genomic variations, a genetic variation. Mainly they consider the single nucleotide polymorphism, and they are associated with these different individuals with the type 2 diabetes, coronary artery diseases, prostate, breast cancer, rheumatoid arthritis, inflammatory bowel disease, and number of mental illnesses. They associate with that, so that has given the strong point for JIVA studies. This is beginning for the JIVA studies, means genome-wide association studies of taking one gene or two genes and studying for the thousands of individuals belonging to that category of the diseases also. So they try to put forth and get an evidence that this variation is responsible for this kind of diseases. Then the the other quantitative scientists also, the technology is again they well developed. Then they say that 270 is not sufficient. Let's go for 1000 genome project. They started 2008 and completed within 3 years, that 2010, and they concentrated major genomic variations of the single nuclear polymorphism, copy number variation, and insertion deletion. These three the variation they found it, and they wanted to create complete catalog of these variations and ask in the secondary one like so by making all of them together to make a reference genome sequence. What do you mean by reference genome sequence? A standard genome sequence where all the variations are there. When you have new individual genome sequence, you can compare that. Okay, this is matching with that. This is not matching. A new variation is there. So to build a reference genome sequence, the 1000 genome project helped 2008 to 2009, and also the as a population specific mutations are there, or what mutations, the recombinations are causing by this variations. They started this 1000 genome project, and they included the populations for the 1000 genome, about 12 populations. They included of different the Tokyo population here, Nigeria, Yoruba, and Chinese Beijing population, Ottawa, Europe population, the Kenya, and Kenya different population, and like Italy, and our Indian population here, Gujarati Indians, the Houston were there, they have been selected about 867 members, they selected for this 1000 genome project. And now we look for the compare our, if at all if we are taking the sequence, we will compare with this 1000 genome project, specifically Gujarati Indians also, many of them are did the 1000 genome project, and later they made a catalog. Now we have the human genome reference sequence. Maybe I am going to tell you later also. That's the updated one we have. So it's due anytime due to review that, renew that the 38 HG 19 or 38 of this. Later in the UK, one of the Prime Minister of the UK, he started that because the death of 1,60,000 people are died in UK in 2012. Record says that so he has started asking questions that in 2011 the 1,60,000 people died of the cancer. He put a lot of money and made the government money for the Genomics England company, and they had the major role to create an ethical, transparent program based on consent and bring about the benefit to the patients. They sequence and to the new scientific discovery to identify the genomic variations, and this David Cameron, then the Prime Minister of the 2012 Prime Minister, he started this, gave money, government money to the private company, and London 2012 Olympics, whatever the money earned, they gave to this to start this also, and systematically for 1,000 genome, 1 lakh genome, 70,000 patients family, and 21, all the data they put forth. Many people involved. They completed this 2018, and the outcome is they identify new variations, they identify cancer-specific genes, the variations, and then now they are put into the medicine for individual cancers. I think, I'm not mentioned. So, 2018 onwards, they identified the variations of the genes responsible for each kind of cancer, and now they are in the treatment. So, UK, the globally is rich in understanding the genomics for the treatment of the cancer patients. 2016, the Genome Asia, when the UK people or the US people are done that, why not the Asian people? They started, they launched 2016, the 1 lakh, 1 lakh or 100K individuals from Asian, like the Singapore University, and Macrogen from South Korea, and Genentech from US and India. And this company joined together, started the collective the Asian, and they did the genome sequencing. But so far, to my record, they have not been announced that also. Announced completely? Partly. Yeah, first phase they announced, but maybe they are holding some of the important the gene, culprit gene, to get the benefit of making their own patent and one kind of the kind of drugs also. They partly announced, but not completely they announced that also. Lately, we have opened up the Indian scientists, the CSR spent money. I think Sami Brahmachari is there. He has initiated some huge money for 20,000 Indian genome from the CSR, and they made the Institute of Genomics and Integrative Biology and CCMB, and they asked, they put forth several objectives to at least 20,000 Indian genome for the rare genome. Can be a diverse ethnic groups they taken, they wanted to do it, but still it's not been completely announced. It's holding that maybe their own group they know that, but not completely announced that. On the cost effective, the genetic test and so on, they're making up that. But I did not say that completely they put it in that website. So still they are working, and also some new information they're getting. Rare genetic diseases, they're looking for the variants. Putting all the genome, whatever they published in the public domain, they made the Genome Reference Consortium Human Build. We call it GRCH38. They compile all the reference genome for the global data, including Chinese and USA data. Whatever the variations were there, they added that. The one group with the B37, they have that one small group, and HG19 and GRCH38. All that say there are different databases are there. So that databases tells us about the reference genome so far, that the references are given that this is the one they make that the reference. Whenever you have a new genome sequences or any individual sequencer, we have to go for this reference genome sequences. So with this they identified, this consortium identified new 210 MB gaps, unknown 151 MB, and computationally simulated 59 MB sequences, which constitute about 6.9, 6.7 sequences. But when they announced 2003, the heterochromatic region, some of the gaps are not there, but with the NSUS article they said 99% they completed, but some of the regions were not being revealed, and that with the new technology to understand the repeated genome sequence that they made it, that is 2022. Though they have 2003 where that it took about 20 years to complete some of the gaps there. And also 2022, kilometer DLO consortium was started, and they also did completed some of the autosomes and the X chromosome, except ribosomal DNA array, they completed. That means these are the ones giving good data or near to accurate data to make the genome is free from the errors there. So this they found about 3.7 million new SNPs there. And March last year, May 23rd, 99% accurate genome sequences, they have taken about 17 phase of 700 haplotypes and 350 individuals, 750 haplotypes they sequenced, they made that and different new duplications and the variations they added. Finally they said that error rate in the genome's reference genome sequence is reduced 1 in 2 lakh bases. So earlier the more of error is 1 base in the 2 lakhs bases. The updated of this human pan-genome reference consortium, they are updated also. Last year, February 28th, in the Science Day, they announced that 2024, the 10,000 genome project was initiated. Earlier, they initiated by CSIR. So we have different small country, different opinion, different group and scientific group. They started, DBT has started, they wanted to fund this for the 10,000 Indian genome, and the objective is to develop predictive diagnostic indicators for several high priority diseases and other uncommon genetic disorders. The CSI group also have rare diseases, but they are also different terminology they use. They made it. I'm not going to comment any other thing. Then another group has started, and they create a reference database for whole genome sequence out of India. This is very, very important for Indian scientists to have our own reference genome sequence comparing with the other Western data. This is very essential. It's needed for any of the upcoming genome scientists. This is going to be good. So DBT is funded, and the first phase, the second phase, they wanted to concentrate with major diseases like cardiovascular diseases, mental illness, and cancer, and that is the proposed. And also they launched the Indian biological data centers. This is very, very important for us to compare our own data, where you stands our South Indian population or individual, wherever the sequence are there, but launch that, but still having many objectives of that, to have a customized drug, develop personalized medicine, and for medical counseling or many of the diagnostic method, to improve diagnostic method, to identify the susceptible genes for the infectious diseases. They launched that, and still we need to get the complete databases, or the databases announced, and the other people can be utilize that also. So far, what I have just monotonously I said that different genome project globally, they made their own objectives, and some of the data have been released, but ultimately now we have the data in the reference genome sequences, any variation, whatever the variations are there, the genomic variations are like any other chromosomal aberrations, are very important, which causes the genetic diseases. Particularly the SNPs and the copy number variations are very, very essential. Maybe next talk I'm going to talk about the genomic variations of this. So if you know, so what? If you know the genome sequences of the millions of people, and what is the good about that? So this will going to helpful for the personalized treatment, individual treatment, personalized, the individual targeted medicine. What is this genomic medicines? Is using the information of the patients and the data to correlate between the genotype and phenotype. Why do you need phenotype? Is a clinical phenotype, and the genotype here, the genomic variation. So can you correlate with this, that this phenotype because of this genomic variation? If you can make some sense out of that, then we can stratify the disease. What is the disease? Is it dominantly inherited, or many factors are inherited, or recessively inherited, or which gene, which pathways is involved in this process, or how this has been affected, and based on that we can select the medications and provide therapy and initiate the preventive measures. If this is the one coming in the family, can you prevent them to have the progeny next, or can you prevent them by giving any kind of medicine? So this is the genomic medicine, knowing about the correct clinical phenotype and genomic sequence data. If this is correct, so now I am telling that correct the clinical data. Clinical symptoms are heterogeneous. We have to have good kind of inclusion, exclusion criteria for an individual to be selected for the sequencing, and designating that this individual has this kind of disease are very, very important. So if clinical diagnosis is wrong, then even genome sequence, whatever you have, it cannot be related. This is also very, very important. And then if you have those clinical phenotype and the genotype data, it will help for the right medicine, for the right patient, and the right dose and right time, which will improve the human welfare. So what is the kind of medicine we are taking? When you go to the doctor now, trial and error medicine. We do have some data, the clinical blood sample data or the serum data, based on that they will give. They will also say there are some infections we have, now another viral infection is coming there. So take this medicine antibiotic, 3 days or 5 days. If it is not come back, then you will change another one. So thinking, but with own experience you will give that, luckily sometimes our immune system is good, so we are curing or we are overcoming this, our immune system, our kind of the food habits, our attitude, our activity is good. So many of them were overcome this disease. Not because of the antibiotic or the medicine, not always, but sometimes it has to be there. I am not denying that. So always we are in the trial and error. So look at the ineffective therapy here. We have several, many of them say BP they have, sugar they have. Hundreds of BP tablets are available. Which tablet, which blocker, sodium channel blocker, potassium channel blocker, or chlorine channel blocker, beta blocker, alpha blocker, which blocker has to be there? The doctors, they try their best, with their own trial and error experience, they will give that something to one blocker. So Americans, by self-medication, they die because of, oh, I know this, the Google search I found this also, they die. So ineffective therapy and wrong drug and also kill that. So many of them are not effective here. They are there, but they are making lot of money. The companies are telling that, even they say that our BP, they were 80 and 120 is not there. Even for Indian, it can have also 130 and also, so you need not worry also. But they say 160, 80, 120, if it go to 130 also in the working time, they see that, oh, something trouble, they give that also. I will tell you, I don't have time. They made me Los Angeles, one of the victims went for the cold. They say that your BP is high, you have to be inspected. They admitted for 1 day in the Los Angeles hospital, just below the lab there. So I was victim for that also. So how much time I have to complete this talk? 10 minutes. So one of the interesting work with my bright, brilliant PhD students, other than their work, we did one case study, some atherosclerosis case study, that was the history of that. June 29, 2030, Times of India, there is an article that the patient, this sample, male 35, had very good lifestyle, no smoking, non-alcoholic, no high cholesterol or poor lifestyle, and he had myocardial infarction, transient ischemic heart attack, and mild stroke attack, and diabetes, and 8 blockages for an artery they found that, and they did with the online American catheterdoscopy, the operation also they said that. After that also, his accumulation of the cholesterol in the arteries were more. So this is the style of the told they had that within 2 months the blockages were more. Somehow he doesn't have any history of that, but they are accumulation. They were giving the Gujarat, this is from Gujarat group, the doctors, these are the medicine they were giving, blocking of this kind of features there. So finally we found out with the paper, we contacted the paper, and the doctor and the hospital finally with MOU got the blood sample for this individual. We are curious about that, we had some money, we got sequenced, that also my Avinash and the Dinesh, they involved in the project, and they sequenced that also. We found many variation. Many of the variations were, other than the PhD work they did in the whole night, the lab, and luckily our vice chancellor gave me on server to process this data, good computer. 13 mutations were we found, out of them are 11 SMPs. There are many, but we found that these are there in the exonic regions, and 2 insertions we found that. Then when you look at that gene, the APO, BAR, STAB, and SOR1 and ITGAM. So these we found out that where exactly the mutation SMP are there, what kind of the substitution and where we identified, and we interacted with the doctors and we explained that these are the genes are defective. They re-described the medicine. These are the medicine that are re-described to attack these 4 defective genes. And for example, LAM2, in the metformin they gave LAM2 was the gene there. If the LAM2 is defective, all the down-regulating genes were about 300 genes were not active there also. So LAM2, activate the LAM2 metformin they given, for example. So the candidates improved for 2 years, they improved, and finally later you know that when the things were good or bad, there is no follow. So we are not able to get even doctors also not able to get. But however, this medicine did work for 2 years, no blockages, it did not come to the hospital. So that's the short story. Now, with the efficient medical care, the screening is more important in the population level, diagnosis and right drug should be given. And what is the future of palliative medicine is that preventive medical care, the screening in the population, already we have databases, okay, this is the one, then we can look for it, and that kind of medicine can give, monitor them, and also we can prevent. Even now, UK 4 years back, they planned that any newborn baby genome sequence will be there given to the mother. That was the plan, but because of the economy, they broke it 3-4 years back, and they stopped it also. Then I think they are going to start. Any newborn baby will have a genome data, so that way they can monitor the whole life that also. So, what is the future of genomics? There are many complex genetic diseases, risks we have to identify, roadblocks we have to identify, signals and therapies has to be done, and comprehensive catalogs for all molecular the variation the lesions are needed, and this to expand the basic science, foundational resources and technology for genomic medicine is very important. So quickly I'll go for it, another 5 minutes. We have only 4 nucleotides, but we do have 4 modified derived nucleotides. We have to remember the students. The cytosine is very vulnerable. It's very vulnerable. This side, methyl group added, one kind of activity. This methylated pharmaceutical group added, another activity. So the Indian scientist Anjana Rao in 2010 only, they made 2 derivatives of the cytosine. Cytosine is a 5-methyl cytosine, that's called as 5th base, a derived base, and the hydroxyl methyl cytosine is the 6th base. These they have been seen in the gene silencing regions, the atopomentanase regions. They identified this one, and they also a lot of other information they said that. Remember the derivative of cytosine by adding methyl or hydroxyl methyl group. They are the 5th and 6th nucleotide. Another in 2011 they moved 2 more bases, derived bases, again cytosine, formyl cytosine or carboxyl cytosine. The 7th and the 8th bases, and these are involved in reprogrammed adult cells, stem cells. Reprogrammed adult stem cells, the formyl means active, methyl is inactivated, and this they have been seen by methylation studies and so on. They could able to identify these 2 more bases, 7 and 8. So cytosine is undergo modifications. It can have methyl or formyl group or carboxyl group or hydroxyl methyl group. 4 different derivatives can be formed in the life. This scientists have identified 5-HTT, which is the regulatory gene of the serotonin. The scientists call them as happy genes. Many a times, for example, faculty they get 3 lakhs and guest faculty get 30,000. So, or the temporary faculty get about. So, even temporary faculty will get 10,000 rupees, he is happy or she is happy. Oh, anyway, I can lead life within that also. Sometimes the faculty get 3 lakhs also. No, my friend is getting more and is getting BMW, I'm not getting, I'm getting this only. Or many of them are even their wife, no, my neighbor got that, I'm not getting. We are more greedy because our serotonin is not regulated. Sometimes people are undergo depression. What they give indirectly a derivative of serotonin or the regular serotonin. Then morning they say, how are you? No, today I'm fresh. I'm okay. Because the gene is been regulated. So the scientists identified happiness gene. So even if you don't know, you can secrete the happiness gene. Be happy. Whatever is there. Your sitting here is the happiest one. Your parents, your grandparents, the whole environment given you opportunity to survive here. Be happy. So, color blindness. 9% of humanity have the color blindness. They are not able to identify the red or green signal. In the signal they go, they look at the neighbors who are there, they push that also. Even though that effect. If none of them are there, they go very fast. Other person they don't might have a green signal. He will or she will come. Then more accidents will be there. Friends, there are correction glasses are available. If you here 9% means that maybe 1 or 2 more, I can find it here also. That if anybody has a problems of the color blindness, you can go to the doctor, write to the doctor, you can have a correction glasses also. So you can save yourself and also other person in this. And hope all of you are aware of Angelina, and she had breast cancer, she had BRCA mutations, her mother also had the BRCA same mutation, her aunt also had the same mutations, and she took a challenge about the genomic medicines of surgically remove the mastectomy, and she is taking a monoclonal antibody for this, and she is surviving, giving challenges, telling that okay, still I am a model, I can. So, how our genomes have evolved from prokaryotes? There is the two transitions were there. First transition was the first eukaryotic transition from the bacteria, that is 5,000, around 5,000 is the bacteria, become 10,000 gene, first eukaryotic one. The second transition is from the first eukaryote to the first vertebrate, from 10,000 gene to the 30,000 gene, who are responsible for duplicating the genome, increase the genome size. There is a duplication of the genome there. And acquiring the function of the genome, we do see in eukaryotic genome. Plants, what kind of plants are they diploid, applied or polyploid? Plants are having polyploid genome. And the genome, if the gene is duplicated, it can, both the gene can work, one gene can be degraded, or one gene can function the same function, another gene can defunction also. This is during the course of evolution, this is the gene evolutions or gene family evolutions are there. And also we have, I hope this terminology you are aware of it. Somebody might have taught you in schools, colleges, pre-university also. Right? There is a lateral gene transfer we see in bacteria, but it's very, very rare. And also we see this kind of lateral gene transfer in eukaryotic system. Mitochondria to chloroplast, chloroplast to mitochondria, chloroplast to the nucleus, nucleus to chloroplast, vice versa. All four directions are there. There are not many evidences are there. Lateral gene transfer also contribute for the genome evolutions. The genome size and genes are not going with the complexity of the individuals. Look at the amoeba, about 67 million base where they predicted. Amoeba, I hope you know, protozoan, the smallest living, the cell organism there, and the protozoan, but the genome size is micronucleus is there, the bigger paramecium there, the protozoan they are bigger one here. Whereas in human about 3 billion haploids are there, but the yeast about 12 million, 12 MB and 6,000 genes, about 2,000 nucleotides for one gene are very well defined. And here the complexity of the organism is not because of the gene or the genome size. So in plant the polyploidy is there. To conclude the whole aspects, the understanding of the genome or genomic studies is very, very important to identify specifically the human susceptible genes. Now people are worried about the new viruses, okay? And it could be a derivative. Maybe I think in the next talk I might have something about COVID-19, the evolution of COVID-19 genome there. Yes. Future vulnerability to diseases and are sensitivity to the certain particular stimulus and so on. We need to understand. So this will going to help us to take care of the populations where the rare diseases are there, prevent prevent those diseases by prenatal screening, and where the in the public domain where the COVID-19 pandemic were able to address. Now we are happy that COVID-19 may come, but still we have our own genome is ready to accept that our genome systems are also. So now, along with that, AI can also be used to understand the genomic variations, and with the critical and the creative human mind, this can be resolved also. So there are many companies are available to make the genetic testing and the drug or genomic medicine, whether we like it or not. In few years, it will be there. They will last like a blood lipid profile. They said that they will last also. Your family is something like related to the HAR, the gene defective breast cancer. You go and test this. It's about 1,000 rupees or 2,000 or 5,000 rupees. You test that also. I will give you medicine. A day will come very soon. Their day will come, and genomic variation understanding, like a lipid profile understanding, is very essential, at least to treat or to understand that also. So, with this, to conclude my talk, though the genetics were being rediscovered 124 years ago, that's 1900, Mendelian principles are rediscovered. Even today, they have more application. We have to teach the students, even different level, not only the students, even the common man should also be aware. Now, everyone say the common man after the COVID, oh, genome sequence, they sit for the sequencing, they said they do not know what are the sequences, but still they are aware. And the mobile phone 10 years back, they were not there. Now everybody is handling that. We are laughing at the people or, the speaker, they are holding the mobile phone also. Now it's an essential commodity now for everybody to communicate also. Like that genome data, genome sequence data, going to be available. It will going to revolutionize the healthcare. When we have a radiology department to scan for the fracture or any kind of thing, why not the genomics lab in the medical colleges? In India, we are very, very late like tube lights. I don't know when the politicians or government or officers are taking up that also there. But already when I worked in two universities, my department was associated with the hospital one. They were in the hospital one. So we have to, there's something, medical school is different. Medical, when you go to a doctor, you have to know. They listen to that. Ramchandra, whatever said is gone above my head. I can't understand. After they talk, they say that what I had to tell. The doctors, many doctors, I address them also. This is situation, they don't make a link. It's a time for the scientists, all this, the learned person to think about this also. Not because I am coming from the genetics department, I started the genetics and genomics department. They had a vision of even 20, 30 years ago. Our university should have that also. I did not get an opportunity, but I got an opportunity to get money from the UGC as a RUSA grant. With the 5 crores, the department has started. Luckily, our Vice Chancellor Generals to agree whatever I say, and that department was started there. So, genetic diseases, many doctors say that, no, genetic disease cannot be. So, tell me, then you will give his own sample or his own medicine to satisfy you, will take his charges there. Genetic diseases are preventable, treatable, and manageable. We are not reached the stage for all the diseases to cure, but we can treat them also. We can manage them also. Therefore, genomic study is inspired thinking take up the challenges, eliminate new hypotheses, affect the society by the views, and it has enormous applications, not only in human, even agriculture, forestry, environment, and the social sciences also it has. Forensic medicine it has. The challenges, the changing society there are personal genomics, personal medicine, the genomics. This is my always say, because my behind my all the students, my PhD students and project students, my mentors and the funding agency, all are supported what I'm studying today here and talking to this. For this, the personal genomic medicine work, the Dinesh and the Avinash, these two members were involved on this work, and others are for a different kind of work. So some of the project students also work with the 1000 genome project. The data we downloaded, we work many kind of questions we asked, we did that also. Even migratory route, maybe in the university where distilled water and electricity were having a problem, you can work on the genomic. People ask me, IIC, no, Dr. Balram said that, Ramachandra, you do this, we give the other material, we will work. Why I have to give to that? My department, my university is fair enough to do that. I had a lot of argument with the ROSA grants there. So the people always wanted to, take away from the university where we have very honest, sincere brain sitting here. We have to make them to do good work and think about that. So with this, my course yesterday, the 6th has started on principle of genetics. It's a 40 episode course, 40 videos of videos are available there, and 40 lessons are there. And if you can click on here, I'm not making any advertisement. It is the government of India spent lakhs of rupees on this course, like this course. Maybe tomorrow I'll go to other courses also. And this is the one you can go to the SWAM portal, and you can free register. You need not pay any money for this. If you want to read that, if you don't like it, leave it there only. But you register. Some of the courses might be interested. You can take that. If at all, if you want to take the credit, 4 credits, you have to take the examination. That is, I think they already mentioned that 25th of May, they have the examination in the first shift in the morning. They have that examination. We made that also. So timeline, unless you timeline, the student may not take that. And now I think there are more than 600, nearly 700 people have registered. And this is the 5th cycle I'm doing this. And people are from Cambridge and Vancouver, they registered, they enjoy, they send the comment to me also. So, like a fun, the study is a fun. So you can register and see some videos, how well I presented or how badly I presented, you send your comment to me also there. So learn the genetics. By mistake, you learn better. Okay, you make mistakes. This is the course what I'm giving now. Next semester I'm giving the human genetics course. That's also same way. That is Government of India, the Minister of Education is supporting that also. So I'll stop it here, and if you have any questions, so I can take up now, and with 5 minutes break, the next, we resume it for the next one.